So this is chapter 13, and it's what we call host-pathogen interactions. So we're going to kind of dive in a little bit and determine, how do these organisms that live on our body surfaces interact with humans? So colonization, infection. Um, Infection is a condition in which a pathogenic microorganisms are able to penetrate host defenses, enter the host tissues, and multiply. A pathological state is the cumulative effects of infection damage. It's a disruption of tissues and organs, and this results in disease. You guys just got done going over an entire unit talking about specific infections throughout the entire body. Now, a disease is any deviation that is from a healthy state. Factors that can cause disease include infections, but we know that disease is not just a state that's caused by infections. It could be genetics, aging, malfunctions of the systems or organs. So there's a lot of things that can contribute to diseases, but we're really focused on the infections aspect. Infectious disease is a disruption of tissues or organs that's caused by microbes or their products. Now we have what's referred to as microbiome, resident biota, normal biota, has many different descriptive terms. This is the large and diverse collection of microbes that live on and in the body. It is also known as resident or indigenous biota or normal flora, or as I just mentioned, microbiome is a term you've heard me use throughout the course. It can include an array of bacteria, fungi, protozoa, and viruses, and these organisms have a profound effect on human biology. Now, the relationships among resident, transient, and disease-causing microbes in the human host is one that's studied a lot because we have trillions of resident microbes, a few of which are pathogenic and a few which are transient, that can be present on our surfaces. Um, the infection is when these potential pathogens and occasion resident microorganisms can actually cause an infection, which then can lead to disease. Depending on the disease, it might actually lead to death. So it's a fine balance between these organisms that live on our surfaces, especially the colonized ones. Pathogens are pathogens. Pathogens are going to make you sick, but we have what are called opportunistic pathogens. Opportunistic pathogens would be our resident microbiota that can cause infections, especially if conditions change. So the Human Microbiome Project, the preliminary results, human cells contain about 21,000 protein encoding genes. Microbes that inhabit humans contain over 8 million protein encoding genes, which is an astronomical number. And a lot of those proteins that are produced by bacteria might actually be beneficial to us as humans, especially on how our overall physiology works and operates. We have a lot of microbes in places we used to think that were sterile, and all healthy people harbor potentially dangerous pathogens, but they tend to live in low numbers. And a lot of the good microbes are really what are keeping a lot of those bad ones in check. So, the importance of gut bacteria, the makeup of your intestinal biota can influence many facets of your overall health. Differences in the gut microbiome have preliminarily been associated with differences in risk for Crohn's disease, obesity, heart disease, asthma, autism, diabetes, and moods. So, there's an old saying that I grew up with when I was going through college and stuff. It was, happy gut, happy person. And there's actually a lot of truth to that. Currently understanding the sites containing normal microbiota, sites definitely known to harbor normal microbiota are the skin and adjacent mucosal membranes, your upper respiratory tract, the GI tract, including the mouth, outer portions of the urethra, which is part of the urogenital tract, your external genitalia, the vaginal tract in females, external ear canal, and external eye, the lids and the conjunctiva. And again, you should know these. These are all things that we covered in Unit 3. Additional sites now thought to harbor at least some normal microbiota, or at least their DNA, are the lungs, lower respiratory tract. They've been able to identify some DNA down there. Bladder and urine, breast milk, amniotic fluids, and fetus. Sites in which DNA from microbiota has been detected is in the brain and bloodstream, but we do not see any really noticeable organisms in those sites. So how do you acquire your microbiota? The benefits of normal biota influence the development of organs. They prevent the overgrowth of harmful microorganisms. There's a term that's used, which is microbial antagonism. This is the general antagonistic effect of good microbes have against the intruder microorganisms. And the way I like to explain this is picture that you have only so much nutrients available. And the good normal flora that lives on those surfaces, that's their home. And they're going to defend that home. And, they're going to defend it, especially against these bad guys that might tend to absorb a lot of those nutrients. They're going to keep those numbers low. So microbial antagonism is a way in which we can keep our normal organisms at lower numbers. Microbes in a steady established relationship are unlikely to be displaced by incoming microbes. So your good gut bacteria will definitely defend against new and intruding bad ones. Now, factors that can weaken the host defenses and increase susceptibility to infection: age. You might have heard me talk about through the chapters in Unit 3 that very young and very old are more susceptible to infections. We have genetic defects in immunity and acquired defects such as AIDS, which is a acquired immune deficiency syndrome. This is the result of an HIV infection that is not managed very well. Pregnancy, surgery and organ transplants, especially if you're on chemotherapeutic immunosuppressive drugs as a result of the organ transplant. By their definition, immunosuppressive drugs are suppressing the immune system. Underlying diseases, cancer, liver malfunctions, diabetes, these are all immune suppressive conditions. And just, physical and mental stress. If you're stressed, your immune status is going to be suppressed. And infections breed infections, other infections. So endogenous infections, these are caused by biota that are already in the body. They can occur when normal biota is introduced to a site that was previously sterile. For example, E. coli is normal biota that we find in the gut. However, if it enters into the urinary tract, that results in a UTI. So an endogenous infection is one that arose from our organisms that are already living inside the body. So when does it start? A growing number of doctors and scientists believe fetuses are seeded with normal microbiota in utero. These microbes are important for healthy full-term pregnancies and healthy newborns. We know exposure occurs during birth when the baby becomes colonized with the mother's vaginal biota. So as they're passing through the vaginal tract, they are picking up everything that the mother has colonized within the vaginal tract. Breast milk contains around 600 species of bacteria and sugars that babies cannot digest. Sugars used by healthy gut bacteria. Breast milk may be necessary for maintaining a healthy gut microbiome within the baby. And, here, again, where do we get our colonization? Where does it start? In utero, at birth, through breast milk, caregivers, handling a newborn. And then just in the environment, if you have a baby that's crawling around on the floor or crawling around the environment, they're going to pick things up. One thing that you need to do for your child is to let them lick things, let them put things in their mouth, let them be exposed to these things. It's good for their overall immune system. So, life on humans, sites that contain well-established biota and representative examples. Again, a lot of these you've seen already from Unit 3. This is sites where we have, normal microbiota, and you should be sort of familiar with what types of bacteria are found in each of those. Skin, GI tract, oral cavity, the intestinal tract, respiratory tract, especially in the nose, throat, very little in the lungs, vaginal tract in females, urinary tract. Those are all sites where we find these organisms. Now, the progression of an infection, a pathogen is a microbe whose relationship with its host is parasitic, and this results in infection and disease. True pathogens are capable of causing disease in healthy persons with normal immune systems. Opportunistic infections, on the other hand, these cause diseases when the host defenses are compromised, when they become established in part of the body that is not natural to them. Opportunistic infections or opportunistic pathogens are typically part of your normal microbiota. But what's happened is the environment has changed. The conditions that were governing that relationship have changed. We have what are referred to as biosafety levels. This is a system of biosafety categories that was adopted by the Centers for Disease Control and Prevention. It's based on the general degree of pathogenicity and relative danger in handling of these pathogens. The primary biosafety levels and agents of disease, we have 4. Biosafety level 1, low infection hazard. Microbes are generally not considered pathogens and will not colonize the bodies of healthy persons. This, you really do not need to have any special precautions. Biosafety level 2, these are agents with moderate potential to infect. Class 2 pathogens can cause disease in healthy people, but be contained with proper facilities. Most pathogens belong to class 2. This requires lab coats, gloves, eye goggles when you're handling these organisms. Biosafety level 3, agents can cause severe or lethal disease, especially when inhaled. And a lot of BSL-3 organisms are aerosolized. Coronavirus would be considered a class 3. Flu is considered a class 3. Class 3 microorganisms, again, can be passed through the air. When you're operating with this, you want to have all of the precautions that you have in category 2, along with potentially using various containment units, working underneath the hood that has a negative pressure so that none of the agents can get out or into the air. Make sure you have filtration systems in place, so on and so forth. Biosafety level 4, these agents are highly virulent microbes that pose extreme risk for morbidity and mortality when inhaled in droplet or aerosol. Most are exotic viruses that we really don't see a lot of. You need to have all of level 3 practices in place, plus facilities must be isolated with very controlled access. Clothing changes and showers required for all people entering and leaving, and materials must be autoclaved or fumigated prior to entering and leaving the lab. Biosafety level 4 labs are highly restricted in the United States. Most companies do not have a biosafety level 4 lab, and actually most universities do not. The ones that do are in tight regulation with the federal government. And most biosafety 4 labs are actually run by the federal government. And they are usually in remote areas. Just alone in the city of Philadelphia, there is actually a law where you cannot have a biosafety level lab 4 in the city of Philadelphia. And most cities actually have that as a law because you don't want to put these labs in very populated areas. The only city that I know of would be Atlanta, and that's where the Centers for Disease Control is located. Okay, now virulence. Virulence is the degree of pathogenicity. It's indicated by the microbe's ability to establish itself in a host and its ability to cause damage. A virulence factor is any characteristic or structure of the microbe that contributes to toxin production or induction of an injurious host response. Now, the size of the inoculum can play a very important role. We have what's referred to as an infectious dose. This is the minimum number of microbes that's required for an infection to proceed. It's determined experimentally for many microbes. Microbes with smaller infectious doses have greater virulence. So what we're going to get into now is what steps are involved when a microbe causes disease in a host. And there's 5 steps that are involved in what we call pathogenesis. That's what this is, how a microbe can cause disease. Pathogenesis is how microbes can cause disease. And there's 5 steps. 1, you have to enter. 2, you have to establish an attachment to the host. 3rd, you have to survive host defenses. 4th, you have to cause disease. That's the whole purpose of a pathogen. And then 5 is exiting the host. In recording 2, we'll pick up with this particular topic. So this will be the end of recording 1.